So I'm going to talk to you about the impact of poultry intensification, particularly on antimicrobial exposure and bacterial risks to human health. Now, we've already had an introduction to the hub. I'm not going to go through it again. I'll highlight that we're focusing here on the microbiological aspect, so that's specific bacteria, microbiomes, and antimicrobials. Now, Guillaume touched upon the different experimental designs we've had in this study. I'm going to give you some results from the first cross-sectional study, as a lot of the more recent longitudinal data is still being analysed. So I'll start off with a case study talking about antimicrobial residues. We want to talk about the possible links to antimicrobial resistance, talk about gut microbiomes, and I'll finish up, assuming I have some time, with some specific examples related to Campylobacter. So we start off by thinking about antimicrobial use, and in particular meat residues. Now, in poultry, antimicrobial use remains common. It can be used therapeutically, but in some countries it's also used prophylactically. And of course, the different antimicrobials available for use are categorized different ways, but we're using the AMEG system, where we're looking specifically at high category, so category A, those are antimicrobials that should be avoided and not used in veterinary practice, all the way through to category D, at the bottom, where they might be used more widely, but they should be used prudently. We're looking at examples here of sulfonamides, tetracyclines, beta-lactams. And we're particularly interested in the risks that are associated with using these antimicrobials and selection for resistance. Now, in the hub, we've addressed this in multiple different strands. Through an epidemiological questionnaire, we collected data on antimicrobial use, so the drugs farmers understand or believe they're using with their poultry. Our social science colleagues have conducted interviews and workshops to again explore the use and the understanding of the antimicrobials with the system. And we've also collected biological samples, and that's what I'm really going to be focusing on. So we collected meat samples, where you can measure the residues of antimicrobials that are present, particularly those that are relevant to food safety. And we've also collected other samples, for example, gut contents, so we can look at specific bacteria or microbiomes and the impact in the same chicken. And as I said, I'm going to touch on several case studies. They're all going to be quite brief, but if you look at the bottom of each slide, you should see the people who've led and provided the slides or the data for these. So we'll start off thinking about meat antimicrobial residues. The work here has utilised EU reference lab protocols, and this is a liquid chromatography mass spec approach. And it allows us to screen 69 different antimicrobials, and we can assess them at maximum residue or maximal residue limits. And so that's the limits at which the residues may be permitted in food for human consumption. And so there's a complex chain system. The samples are collected in all of our hub field sites. They're frozen immediately. They're transferred to Singapore, where we've developed this at Nanyang Technological University, taken it through this pipeline until we can at the end identify the presence of antimicrobials in those meat samples. And the output from these may be a sample that is non-compliant or a result that is non-compliant. So if it's a result, it's where one antimicrobial exceeds the MRL limit, and a sample is where the sample contains one or more antimicrobials that exceed that limit. Now, there's a lot of information here. I'm not planning on talking through all of it, but what it highlights is the complexity of the antimicrobial residues that have been detected. So if we just look at the example at the top of Vietnam, see that 14 samples were considered non-compliant, and these were mixed across farms, and of course that could be during the production cycle, so it may not be a problem. It could be before withdrawal, also at markets or slaughterhouses. Now, the sharp-eyed among you will see there's more non-compliant results than samples. Of course, that means that one sample had more than one non-compliant result. And if you look at the list of antimicrobials found, perhaps I'll just highlight the ones in the B groups, as those are the ones of the most importance or significance. So, norfloxacin, ciprofloxacin, enrofloxacin are present. What's interesting is that a lot of this did not align very well with the recorded antimicrobial use from farmers. So it's not necessarily what they believe they put into their chickens. So there's a poor correlation. Now, the value is, because we've used a similar method to the EU system, we can actually compare those results. And if you look at the prevalence of residues at endpoints, and we're talking about endpoints here because that's the point at which meat actually enters the human food chain, you can see there's fairly high occurrence of residues. So more than 6% in Vietnam, nearly 9% in Bangladesh, nearly 4% in Gujarat. And these compare to figures from Europe, annually about 0.03 to 0.2%. But of course, remember, we're monitoring here, so there's a lot of pressure on that use. But it does indicate both direct and indirect risks to human health, and also implications for international trade. So if we recognise that we can find antimicrobial residues, what impact does that actually have on the presence of antimicrobial resistance genes? So to do this, we looked at the intestinal samples taken from the same birds used for the meat analysis, and we used the next generation sequencing approach to detect the presence of antimicrobial resistance genes, or ARGs. So we used total genomic DNA extracted from cecal contents. And in total, this targeted 487 ARGs, and we refer to this as the resistome. So we used intestinal samples from chickens collected in Bangladesh, 2 regions in India, Gujarat and Tamil Nadu, and also Vietnam. And in the initial data, they detected 124 different antimicrobial resistance genes that relate to 19 different antimicrobial classes. I know this graph is quite small, hard to see, but it just gives you an indication. So you can see at the top, aminoglycosides were the ones with the most common ARGs found. But we noted at this point that there was a mix of intrinsic and acquired resistance genes. And by intrinsic resistance, I mean ARGs that will be present in a bacteria without the development of resistance. So it doesn't have a mutation, it's not gained it by transmission from another bacterium. It's something that you expect to have there. So, of course, our interest really is about acquired resistance, and that's the flow of antimicrobial resistance genes. Now, to talk about this, I'll just step back for a minute. So, some of the earlier work we've been doing has been looking at microbial populations in chickens. And we've actually found, if you take, for example, a microbiome sequencing type approach, you can see that there's 3 distinct, albeit overlapping, types of bacterial populations you'd find in a chicken seeker. You can see an example illustrated here. And these differences are primarily explained by geography and husbandry, not necessarily chicken breed. And as an aside, it's interesting to note that some of these are more likely to have Campylobacter than others. Now, what's interesting is when you look at that resistome data that we generated, referred to in the previous slide, you again see 3 different clusters or types of resistome. And I'm sure you won't be surprised when I tell you that actually these correlate very closely with those enterotypes. So what this suggests is that actually most of the antimicrobial resistance genes we see are part of the intrinsic resistome. But the outliers, the ones that buck the trend and don't fit with the enterotypes, they're the ones that may be acquired. Now, this is a work in progress, so we don't have final data yet. We're looking very closely at associations between reported antimicrobial use and detected antimicrobial residues, and to see if that affects the carriage of different antimicrobial resistant genes. But we can do a comparative analysis where we're looking again at those resistomes, and we cluster them this time by region. Now, it's quite a brightly coloured slide, but what you can see here in each of these brightly coloured bars is an antimicrobial resistance gene that is specifically defines a region, so it may be over or underrepresented there. And you can see there's genes from Vietnam, Tamil Nadu, Gujarat, Bangladesh. And as we explore these further, we hope to see if these are influenced by the use of antimicrobial drugs. So having discovered that there's lots of acquired antimicrobial resistance genes, we're interested to know where they are and how easily can they be transferred. So, of course, if they're present on plasmids, we know plasmids can be transferred between bacteria very easily. They can also be integrated into chromosomes. Now, our concern is if antimicrobial resistance genes present in commensals may be transferred to pathogens, and of course that can impact on the outcome of infections in humans and poor outcomes for treatment. So the presence of these genes in zoonotic pathogens is a particular concern. Now here we've started to use a different sequencing approach, Oxford nanopore sequencing, and we're sequencing whole bacterial populations. And this allows us to identify plasmids within those populations, and you can also construct the genomes of bacteria, particularly the more numerous bacteria in those populations. Now this is at early stages. Actually, the slide is already out of date. There's been a lot of sequencing in the last couple of weeks. But in the pilot, looking at just 15 samples, so just 15 metagenomes, all from Gujarat, we found nearly 4,000 putative plasmids. If you look at the map below, you can see everywhere there's a dot is a hit where a similar plasmid has been detected before. And nearly 500 of them had been reported before, but that tells us that a very large number are actually novel. So they've not been detected, possibly because they're in commensals or unculturable bacteria. But of course, our primary interest, when you look at the content of those plasmids, yes, of course, you can see there are lots of antibiotic resistance genes here. Examples of aminoglycosides, tetracyclines, lots of those indeed. Tetracyclines again down there, lincosamides. Now, just to conclude, I'm going to look at two specific examples related to specific bacteria, in this case, Campylobacter. So the first is noted that Campylobacter is, of course, a major source of human food poisoning. In the hub, we sequenced more than 500 isolates. I heard today, actually, it's more than 600 now. And when you compare the sequencing of these genomes, as you can see in this tree, anything that's got a pink inner circle is Campylobacter coli. Anything with a blue inner circle is Campylobacter jejuni, both of which are zoonotic, but Campylobacter jejuni perhaps is more commonly a problem in humans. But you can see from this that in some cases the geographic clustering, so if you look at the outer part of this ring, you can see there's areas where there are red bars and green bars. They relate to Bangladesh or Vietnam. But if you look at Campylobacter jejuni, actually there's less evidence of clustering. They're more mixed isolates. And the outer circle of the little black dots, those are new sequence types that have been reported for the first time in the Poultry Hub. Just to highlight, a lot of the sequencing has been done at the London School of Hygiene and Tropical Medicine. And of course, when you look within these genomes, then yes, of course, you can detect antimicrobial resistance genes, and in many cases, large numbers of them. I should say this is not proportional, this is a total number. There are more genomes from Vietnam, so that's partly why they're overrepresented here. And to finish, we're also interested in the specific risk posed by Campylobacter. And so quite a large study, been working in Tamil Nadu in India, worked with a large cohort of children, and they managed to recruit more than 530 children to the study. They're between 6 and 59 months of age. This is, as I say, based in Tamil Nadu. They had weekly surveillance here for diarrhoea, and there was also sequel samples collected from chickens, either reared in the homes of those children or local to them. There were then selective culture used to detect the presence of potential pathogens here. You can see the results of the culture here. So Eromonas was most commonly isolated, not necessarily a problem from chickens, but Campylobacter was in second place. If you look at the build-up of the Campylobacter in the humans, you can see actually the majority were Campylobacter jejuni. There was a smaller proportion of Campylobacter coli, and actually several other Campylobacter species were also detected. What's interesting is if you look at chickens, nearly 50% of the chickens were also carrying culturable Campylobacter. When you look at that population of Campylobacter, nearly 70% of those were Campylobacter jejuni, and about a third of them were Campylobacter coli. So a very high occurrence of potentially zoonotic Campylobacter. And so if you compare the occurrence of these bacteria to the actual amount of sampling that was done, so we're looking at the incidence rate here of diarrhoea associated with the presence of Campylobacter per 1,000 child years. And so that's a measure of the density of the sampling that was undertaken. And then when you compare that with whether a child was likely to be exposed to a chicken or not exposed to a chicken, then you can actually see there's a far higher incidence rate in those children that were exposed to chickens. There's an incidence rate ratio of 3.6 and an attributable risk of nearly 73%. So to conclude, there's very strong evidence of high levels of antimicrobial residues in meat from all of the countries we've sampled. So this isn't necessarily a surprise, as there's not currently direct or regular monitoring for it. Of course, in the European Union, we do have that regular monitoring, and that probably drives reductions in use. But we've also found there are limitations in the questionnaires we use to confirm the use of antimicrobials. So maybe farmers were not understanding what was being put in, maybe it was coming in the feed already, or maybe it was coming from the environment. We're now exploring the epidemiological links between the presence of both antimicrobial residues with phenotypic resistance, so that's culturing bacteria, assessing whether they are resistant, or whether they're carrying the genes that confer resistance. We found a very distinct regional variation in acquired antimicrobial resistance genes, and perhaps most notably, we found a very high occurrence of plasmids in the enteric microbiota taken from chickens, and these include numerous antimicrobial resistance genes. So, of course, these are a threat of transmission to zoonotic bacteria, through the environment, to water sources, and elsewhere. We found there's considerable regional diversity in the presence of Campylobacter genotypes, quite polarised in Campylobacter coli, quite mixed for Campylobacter jejuni, and there's a notable link between exposure to Campylobacter in chickens and childhood diarrhoea. So thank you very much for your attention, and I'll pass you on to Robin.